Okay, great. Well, thank you so much, everybody, for being here. My name is Jeff Sebo, and along with my colleague Becca Franks, I direct the NYU Wild Animal Welfare Program, and we are very, very excited to be hosting Megan Barrett for a talk today about wild insect welfare. So before we get going with the talk, I will tell you just a little bit about the program and what to expect today, and then introduce our speaker, and we can get started. So this talk, as I said, is part of the NYU Wild Animal Welfare Program. This is a research, outreach, and field building program housed within the Department of Environmental Studies, and it examines what wild animals are like, how humans and wild animals interact, and how we can improve our interactions with wild animals at scale, which, as we are about to learn, is especially important and difficult in the context of wild invertebrates and especially wild insects. And so we do that work through our research, as well as through public events like this, as well as through field building activities, early career support of various kinds. So if you have interest in that work, interest in this topic, please do go to the NYU Wild Animal Welfare website and sign up for the mailing list. You can check out some of our other programs and centers and curricular opportunities along the way. Now, as far as this event is concerned, we are going to have about 75 minutes to talk about wild insect welfare, and that will be broken down roughly half into a talk and half into audience Q&A. So we will start by hearing Megan give a talk about the topic, and then the remainder of the session will be me relaying your questions and comments and objections and so on and so forth to Megan. And so at any point during the event, if you find that you do have a question or comment or objection, etc., you can go ahead and enter that in the Q&A tab at the bottom of your screen on Zoom, not the chat tab. You can enter it in the Q&A tab at the bottom of your screen. And then you can also engage with other questions. And then when the time comes for discussion, I can read those out to Megan and we can have a conversation about them. Okay, now, with all of that in mind, I am really happy to be able to introduce our speaker today. Megan Barrett is an assistant professor of biology at Indiana University, Indianapolis, where she researches insect neuroscience and farmed insect welfare. She is also the founding director of the Insect Welfare Research Society, a wonderful organization, and she earned her PhD in biology and MS in education from Drexel University in 2022, working on the neurobiology and thermal physiology of solitary desert bees. She was a National Science Foundation postdoctoral fellow at California State University Dominguez Hills from 2022 to 2023. And Barrett has published over 40 articles and book chapters about insect neuroscience, physiology, and ethics. And her work is already, despite her only having graduated a couple of years ago, quite influential and very important. And we are very happy to have her here today. So Megan, whenever you are ready, thank you for being here. Feel free to share your screen and get started with the talk.
Great. Thank you so much for having me. So let me just pull up these slides. So I'm really excited to be here today. Hopefully those slides just appeared for you. And so, yeah, we're going to be talking about wild insect welfare. And as the bio kind of suggested, my research program is actually mostly focused on farmed insect welfare. So I primarily research black soldier flies and yellow mealworms. But I have worked on wild insects in the past, like the solitary desert bees I worked on for my dissertation, and it's a topic that I wish I had more time to think about. So I'm excited to talk with you about this today and get your thoughts on some of my very early thoughts on this subject. So this is what I anticipate I'm going to share with you today. I'm going to briefly do something I do at kind of the beginning of every one of my talks, which is an introduction to insects and the evidence for insect sentience, and that's to make sure we're all on the same page about what animals we're actually discussing and what the evidence currently is for whether or not they're sentient or have experiences. And then I'm going to get into kind of the more speculative piece, which is about wild insect welfare challenges. I'm going to walk you through kind of the scale we could be looking at, some of the harms that might be faced by wild insects, and whether or not we have any sense of whether we could mitigate them. And finally, one thing that I think gets overlooked are springtails and mites, and so I'm going to very briefly end with a note on those animals as well. Okay, so to start out, let's just make sure we're all on the same page about the animals we're talking about, which are the insects. So insects are invertebrates. Invertebrates are the majority of our animal diversity in the world. So there is the chordates, that's where we are located, and insects are all the way up there in the arthropods at the top of the screen. And so if we zoom in on arthropods, what that will tell us is that things like this, by the way, the little earthworm that just appeared on your screen, not an insect, right? Not in the arthropods. So some animals, some people think are insects, are not. When we zoom in on the arthropods, they're going to be the majority of invertebrate diversity. And there are many arthropods that people also think are insects that are not insects. And so I want to get clarity on that too. So one group, for example, are the spiders. Spiders are arthropods, but they are chelicerates, the most ancestral group of arthropods, lumped in with things like ticks, mites, horseshoe crabs, and scorpions. We also have things like millipedes and centipedes, although lovely. They are also other groups of arthropods. They're in the myriapods. Instead, our insects are all the way up here again at the top of the screen in the hexapods. So there are some non-insect hexapods that are very ancestral, like the springtails I'll tell you about at the end of the talk today. But all of our insects are going to be up in that group, from our weevils, our termites, our bees, ants, wasps, sawflies, regular flies, etc., are all clustered up there at the top. And that does mean that even one other very common terrestrial arthropod that people think when they think insects is not an insect, which are your roly-polies or your pill bugs. Those are actually within your decapod crustaceans, within your crustacean group instead. So you may already think, oh, a lot of things I thought were insects are not insects if you're not an insect specialist. But even though there are many things we think of as insects that are not insects, there are also many more insects than you probably think. So there are a million described species of insects. There are 5.5 million species estimated to exist, and that covers absolutely tremendous variation in lifespan, in behavior, in cognition, in physiology, and thus in kind of the welfare needs of these organisms and the way they live their lives. And so when we think of the term insects, we should think that the only thing those animals really share in common are going to be 6 legs at the adult life stage and some other morphological characteristics. And on every other axis, we should think that they vary. And you can see an example of just the incredible diversity that we have even within just the ants here on this slide with those ant faces. So what I like to do is run through a few myths about insects that I think causes people to have these intuitions that insects are unlikely to be sentience candidates. So one of those is just a size bias where people think, well, insects are really small, small things probably aren't sentient. But that isn't actually true of this entire group. So there are insects that can be really big. In fact, there are insects that are bigger than mice and equivalent to female rats in weight. I've put a couple of examples of giant insects here on this slide: giant bees, giant beetles, the lord house stick insect, the giant weta. Insects can get very, very big. And that means that their brains can also be larger than people expect them to be. When we look at differences in brain mass between vertebrates and insects, we see that actually the largest insect brain that has been studied to date is larger than the smallest vertebrate brain that has been studied to date. So the smallest vertebrate brain is the Algerian sand gecko. It weighs in at 11.3 milligrams. And one of the wasps that has been studied has a brain that weighs at 11.7 milligrams. And keep in mind that that wasp is not anywhere close to the largest of the insects like I just showed you on the last slide. That's a 0.55 gram wasp. And we know that there are insects that go all the way up to 200 grams in body mass. Um, if you're somebody who is a little bit more suspicious of geckos for some reason and you want to just compare things to mammals, the differences between insects and mammals are actually also not that big. The smallest mammal brain is the Etruscan shrew. It weighs in at 64.4 milligrams, so that's 5.5 times bigger than the largest insect that, again, has been studied to date. And neuron numbers tells us a very similar story. Same number of neurons, actually, between the Algerian sand gecko and the wasp. And the smallest mammal that has been studied to date so far is the naked mole rat at 27 million neurons. So probably closer than you might have initially anticipated in terms of computational mass or neuron number in those brains. And those brains are more complex than people have expected them to be. And that includes entomologists, actually. We're sort of becoming slowly famous in my mind for thinking that insect brains are going to be less complicated than they are. So one thing that we think may be important for producing subjective experience in mammals is direct connections between sensory integrated brain regions. And it was posited that that was something that insects lacked, and therefore they might not be sentience candidates. But in 2020, when we completed the first connectome of the adult melanogaster fruit fly brain, we found direct connections between their sensory integrated brain regions, the mushroom body and the central complex. So more complex than we might have initially expected them to be. And this complexity allows insects to live very diverse, very sophisticated kinds of lives. I've put examples on this slide of some of my favorite things that insects do. Hopefully, at least one of them is kind of a new thing for you. And again, these aren't true of all insects, they're just true of particular kinds of insects to give you a sense of the real diversity in behavior in this animal group. A couple of my favorite examples, one would be monogamous pair bonding for up to 20 years in termites, where queens and kings will pair off together and kind of run their colony and take care of their offspring together for 20 years in their nest. We have rescue behaviors and wound treatment behaviors in ants. And in fact, it was recently discovered in a species of carpenter ant found in Florida that they actually are the only other animal species besides humans that engage in medically relevant amputations. So if a nestmate comes back with an injury to one part of their leg or a different part of their leg, the ants will assess the injury location and engage in one of two different treatment behaviors depending on where that location is. If it's a location that is very likely to get infected in a lethal way, they'll actually do an amputation of the leg and then clean the wound. There's facial recognition in paper wasps. So some colonies of paper wasps, every individual has unique facial markings, and they can actually use that information to do a version of transitive inference. So if, Susie beat up me yesterday and I see Jane beating up Susie, I know not to challenge Jane to a fight because if Jane can beat up Susie and Susie can beat up me, Jane can probably beat up me. They can do that kind of thing. And in the fruit fly, we use them to study emotion-like states in humans, including things like chronic pain, anxiety, and depression. So hopefully in the last about 5 minutes, you've learned something new about insects. And all I mean to suggest by kind of bringing us to this point in the presentation is that this is a really diverse group. They have surprising cognitive abilities. They have surprising life history strategies. And we probably have incorrect intuitions about these animals. So we should be really suspicious of our intuitions because if we don't question them, we may make some incorrect assumptions that lead us to incorrect conclusions about insects. If you want to hear me share even more of my favorite insect facts, you can check out this 80,000 Hours podcast episode I did, where I talk at this same pace, but for like 3 hours about all my favorite things about bugs. Okay, so you're like, Megan, that's lovely and wonderful. Thank you so much for telling me all of your favorite facts about insects. But what I really care about for this talk, if it's an insect welfare talk, is whether or not they're sentience candidates. So let's talk about that. So this is a framework style approach that I'm going to present to you for understanding or assessing the plausibility of sentience in a group of animals. And essentially the framework-based approach, what it does is it assesses neurobiological and behavioral proxies that we think are relevant to the plausibility of sentience in a group of animals. And depending on how many of those proxies a particular group of animals has, we may think they're more plausibly sentient or less plausibly sentient. So the framework that's currently on the screen is from Jonathan Birch's group at the London School of Economics. They produced this as part of a report for the UK government in 2021 and used it to assess the evidence for sentience in decapod crustaceans, which are things like crabs and lobsters, and cephalopods, which are your octopuses. And so essentially this framework has 4 neurobiological criteria, which are the first 4, and 4 behavioral criteria, which are the last 4. If you meet all 8 of these criteria, it doesn't mean there's a 100% chance of sentience. It just means the evidence for sentience in that group is very strong. And so we may want to be cautious in our treatment of that group of animals. So this is a precautionary framework approach, not a definitive framework approach. And the other kind of important thing to note about this framework is that although it has been used by Jonathan Birch's group to assess invertebrates, it is actually taxonomically neutral. So you can also use it for vertebrates, and it's based on a framework, the Smith and Boyd framework from 1991, that was initially used just for vertebrates. So it has precedent in the vertebrate literature. So using that framework, we can look at kind of the more accepted invertebrate case first to kind of get our bearings in the use of this framework. So I think many people at this point are willing to extend cephalopods the plausibility of sentience and consciousness. That was true in the Cambridge Declaration on Consciousness in 2012. But also when we look at how they stack up on these 8 criteria, we're seeing that they meet about 7 of 8 of them to a high or very high degree of confidence. And this framework essentially rates that as very strong evidence for sentience in this group of animals. If we want to test our intuitions in another sense, we might think about how we feel about lobsters and crabs. This framework is going to rate decapods with 5 of 8 criteria to a high or very high degree of confidence being fulfilled. And this framework rates that as strong evidence for sentience in those groups. So kind of adjust your expectations according to how you feel about those animals and those numbers. One thing I think is really important to note about the 5 of 8 threshold is just that on the basis of this report and that threshold, the UK government included both of these groups in their animal sentience bill in 2022. So according to the UK government, 5 of 8 was a strong enough amount of evidence for sentience in these animals that they could be considered plausibly sentient legislatively. And so we should think that there's something important, at least in the UK context, about that 5 of 8 threshold. So along with Jonathan Birch's group and some of the authors on that original 2021 report, as well as with Lars Chitka and led by this fantastic now postdoc Matilda Gibbons at UPenn, we all went through and we assessed 6 orders of insects at the adult and the juvenile life stages according to this framework. And this is what we found in our paper that was published in 2022 in Advances in Insect Physiology. So essentially, I'll draw your attention to these 2 orders in particular, the Blatodia and the Diptera. Blatodia are your cockroaches and termites, and Diptera are your flies and mosquitoes. So those 2 orders are meeting 6 of 8 criteria to a high or very high degree of confidence. So 1 less than the cephalopods, 1 more than the decapods. When you look at some of the other groups, like the Coleoptera, which is your beetles, or the Hymenoptera, which is your bees, ants, wasps, and sawflies, you'll see that they meet fewer criteria, but that's largely actually because you'll see boxes filled in with gray that say very low, no research found. So essentially, even for these 6 orders, which of the approximately 30 orders of insects, these are the 6 that are best studied, even in these best studied orders, people are not studying a lot of these criterion in these animals. And so that means that we often couldn't find a single study to point us one way or the other on whether these orders were likely to fulfill these criterion. You'll also note that for both Blatodia and Diptera, the 2 criteria that they don't meet to a high or very high degree of confidence are also both very low, no research found. And so we don't have evidence that they don't fulfill those criterion. We just have no evidence at all one way or the other. The other thing I'll note is just that study came out in 2022. We did a very thorough review of the literature at that time, but of course, science is continuing to progress, and so more research is coming out on this topic. I didn't do a thorough re-review of the literature for this talk, but just from my own reading over the last couple of years, I know those boxes with yellow stars in them have received additional evidence in favor of those animals fulfilling the criterion since we published the 2022 review. And so what this tells us, this fact that at least 2 orders of insects at the adult life stage are meeting 6 of 8 criterion to a very high or high level of confidence, is that there's probably enough evidence for sentience for us to be cautious in our treatment of these animals. And this also appears to be something that, isn't actually that unusual to think among animal behavior researchers and entomologists. So for example, this survey on the left is by Zippel et al. In 2024. They surveyed animal behavior researchers and asked them about which taxon they thought had emotions that would shape the animal's behavior. So in insects, you can see there's 4 different colors. So essentially dark yellow is that no members of the taxon display emotions. Light yellow is that some do. Light green is that most do. And then dark green is all or nearly all do. And so if we add together the some, most, all, or nearly all, 67% of animal behavior researchers ascribe emotions to at least some of the insects. When we talked to entomologists, this was a survey that I conducted in 2024 of entomologists in the Royal Entomological Society and Entomological Society of America mostly. They were 11 times more likely to care about insect welfare than indicate they didn't care about insect welfare at all. So it does seem as though there's some evidence and some consensus starting to suggest that people want to be cautious with these animals. And, in case you need more ideas for, where the field is moving, these are 2 additional documents that kind of demonstrate to us that people are starting to think there's a need to be cautious in our treatment of these animals. The first is, of course, the New York Declaration on Animal Consciousness, which probably everyone at this talk is aware of. But in case you're not, essentially this declaration suggests that sentience is plausible in insects, and as a result, we should be cautious in our treatment of them. It has many signatories, including from the cognitive sciences, from entomology, and more. And the Royal Entomological Society, which is the world's oldest entomological society, actually issued a statement last year on the ethical treatment of insects, in which they again recognize that there is uncertainty about insect sentience, but that it is plausible enough that we should start using practices that minimize the risk of unnecessary harm in our research. Okay, so that's what I've got for you on, introducing you to this group of animals and providing you some of the evidence on the plausibility of sentience in insects at this time. Now let's actually start to move into the wild insect context, starting by talking about the scale. How many insects are there in the wild? So the Smithsonian Institute estimates that there are 10 quintillion insects alive at any moment in time. And I don't know about you, but when I hear the word quintillion, it's sort of meaningless to me because it's such a large number that it has no bearing on anything relevant in my life. And so it's very hard for me to visualize what 10 quintillion insects actually means. So to try and put this in perspective for you, imagine we froze a single second of time, this exact moment, and we just wanted to count all the bugs that were on the planet in that frozen moment of time. So let's imagine we can count 10 bugs a second. We're really fast. How long would it take us to count all the insects at 10 a second that were alive at that moment in time? So if we did that, it would take us 31.68 billion years to count all the insects that had been alive in that frozen moment. So that would mean we'd count from now all the way back to the Big Bang, from the Big Bang all the way back to now, and several billion years into the future to be able to count all the insects alive in that one frozen moment. It is a lot of animals in the wild. And, as you might expect if you work on wild animal welfare, there are going to be many of the same concerns that affect other groups of wild animals that are also likely to affect wild insects. So we've got categories of natural concerns, things like starvation, predation, adverse environmental conditions, like increased temperatures and the like. And we have anthropogenic concerns. That would be things like pesticides, harvesting for food consumption or shellac or other purposes, the effects of pollution, habitat loss that leads to, the loss of resources that are important for that species behaviorally or nutritionally. All of those are going to be things that can be concerns for wild insects. I'm going to, at this point, take a kind of case study approach to looking at three different um, things that might be concerning that wild insects face. And I'm just going to kind of speculate about those three things and what we might think the welfare harms could look like, what the scale of those welfare harms could be, and ways that we could think about maybe trying to mitigate them. I'm going to start by talking about pesticides on cropland. So essentially, we've got a lot of cropland in the world. About 38% of land globally is used for agriculture, and about a third of that is used as cropland. So about 16.5 trillion meters squared. We've got very few estimates of how many insects there should be per meter squared in a soil of cropland. So I used some rough estimates from other kinds of ecosystems. Let's assume about 1,000 to 3,000 individuals per meter squared of soil-dwelling insects. Also, I'm saying insects-ish because often people actually do include spiders and some other non-springtail and non-mite groups in these estimates, and I was not able to separate those out. So insects-ish, we've got about 1,000 to 3,000 individuals per meter squared in the soil. If we multiply all those numbers together, we get a very rough estimate of about 34 quadrillion insects that are going to be on cropland and impacted by pesticides. Now, again, some of that cropland is going to be organic. Some of that cropland is not going to use pesticides as often. There are all sorts of ways we might think that this estimate could be a little bit fuzzy. But let's just say that, 34 quadrillion, give or take a quadrillion or two of insects potentially impacted directly is still a really large number. And so when we think about the potential welfare consequences of pesticides, there's obviously the lethal effect, which I think is what is most immediate to people, the fact that like many of these insects will just die, and many of those deaths could be potentially very painful. Like if it's a neurotoxic pesticide, we could imagine that that is a painful way to go out. But I think we shouldn't ignore the fact that there are also going to be many pesticide applications that result in sublethal effects. That includes sublethal effects for the animals on the cropland, but also sublethal effects for an untold number of animals off the cropland that didn't get accounted for in my 34 quadrillion estimate. That is because when you are spraying these pesticides, as you can see, the wind is going to carry them off of the cropland. When it rains, the water is going to wash them away from the cropland. And so we know that these pesticides can kind of move to areas that are not directly agricultural areas and have effects on animals in those areas as well. So some of the sublethal consequences that we know can happen even just for insects, not talking about, vertebrates and other invertebrates that might be affected by sublethal doses of pesticides. Some of those effects include things like nervous system issues, impaired learning and memory, misfiring motor neurons that lead to different tics, myocardial cell dysfunction, impaired flight, reduced muscle efficiency, developmental issues that can lead to malformation of the cuticle or other body parts, leading to an inability to perform particular behaviors, abnormalities of the reproductive system, impaired immune function that can make it harder for these animals to withstand disease. And, ultimately, many of those can lead to decreased longevity for these animals over time. So both lethal consequences, but also we should not ignore the potential for sublethal effects, both for animals that don't die on the cropland and for animals off the cropland. So, when I started looking into, are there ways that we could try to mitigate this particular welfare harm. I think it's a really complex issue that hasn't received a lot of research, and so I don't want to try and oversimplify it for you here. I guess what I'll say is that a lot of what I actually was reading about was focused on conservation, because of course biodiversity decline is also happening as a result of the over application of pesticides. And it's entirely possible that we could use a lot less pesticides than we do. Reducing the dosage of pesticides, changing the timing of application so that the timing is refined just to the target species, so we're affecting fewer non-target animals, would be one way to potentially reduce the scale of this harm while still meeting other goals like, human nutrition associated with crops. Um, we could also consider potentially using pesticides that are tailored to specific pest species instead of being pesticides that act on all insects. So an example there would be that there are some Bt toxins that are specific to unique orders of insects. And so if you have a crop that is primarily targeted by lepidopteran pests, which are butterflies and moths, you could potentially use a Bt toxin instead of a neurotoxic pesticide that could kill all of the insects and many other animals that are living in that cropland. I think your cost benefit analysis there on welfare grounds is just going to need to factor in that many of those toxins are going to have a much longer and potentially worse death for the target animals. And so you have this question of, more suffering in fewer animals. Does that kind of weigh, how does that weigh out against potentially faster deaths and lesser suffering, but for more animals? I don't really know what to make of the sign of that. I don't think there's good research on it that I've at least seen yet. Again, this isn't my specialty, but I think it's worth looking into whether those pesticides would potentially reduce suffering. Um, Another possible option that some folks are moving towards is using insect repellents instead of insecticides. So here the goal would be to have insects that are coming into, say, oviposit in fields of crops, lay eggs that are going to turn into larvae that will consume those crops, repelling them so they're not laying their eggs in those crop fields instead, potentially not laying eggs at all. That may be a way to, again, reduce suffering. There's a lot of complexity in terms of what pest you're using, what wild resources are available for those pests, whether you're deterring larvae or adults from laying eggs. All of those are going to matter, but certainly something that could be studied more. Um, there's also a question of if there are application methods that could reduce sublethal exposure. So some genetically modified crops, the pesticide is actually sort of produced by the cells of the plant. And so there's less opportunity for runoff or, sometimes less opportunity for that to be carried by the wind into nearby ecological areas, depending on the kind of plant that you're looking at. And so those application methods could substantially reduce sublethal exposure for animals that are not in the agricultural area. And that may be a way, again, to try and mitigate some of these welfare harms. I think ultimately one thing as I was reading up on this topic is, noting that there are both tensions and compatibility between welfare and conservation aims. There may be some cases where those two actually really go hand in hand, and the things that are good for biodiversity are also good for animal welfare. And there may be some cases where those two things are actually in tension with each other, and the things we might think are best for welfare are not as good on conservation grounds. So I think there's a lot more work to be done on humane pesticide development that can go beyond, again, just developing new pesticides and into things like refining dosing schedules, refining timing, all of which could have massive consequences on the welfare of all the target and the non-target animals in those fields. The second case study I want to briefly discuss is about spongy moth outbreaks. So the spongy moth is an invasive species. It was accidentally introduced in North America from someone who hoped to be able to make a new version of the silkworm moth. That happened in 1868 to 1869. They have since spread to, close to 1 million kilometers squared. It's about a quarter of its possible range in North America, and it is still invading new areas. The invasion front spreads about 4 to 10 kilometers each year. It eats a wide variety of trees and shrubs. Usually it is present at really low levels in the environment and actually not noticed by most people. There are probably if you live in a spongy moth area in North America, there are probably spongy moths around you kind of every spring and summer, and you don't really notice. But sometimes environmental conditions are just right for a localized outbreak to occur in an area, and then people definitely notice. So this is what I mean about localized conditions. Essentially, we have some low level that is below outbreaks where natural processes tend to keep the spongy moth population at a relatively low number. We have some ideal weather condition that can help the population grow. And at some point, that ideal weather condition is so perfect that the population reaches outbreak levels. And so we get this spike, this really huge spike in population. That spike usually is a kind of a 3-year cycle from the point where it starts to the point where it begins to drop off precipitously is a 3-year cycle because at some point the population gets too high to survive. So things like diseases will spread and rapidly cause death in the population, density-dependent diseases, and starvation because they just literally eat every piece of foliage in their area will happen. And so a combination of starvation and disease tends to wipe out these animals in the second and third years of that outbreak, bringing it back to a level below what most people consider to be noticeable. So when I was still working with Rethink Priorities, I did a quick case study of an outbreak in Ontario, Canada. And essentially that outbreak, we found that there were about 6.5 to 11 trillion caterpillars on about 6,000 kilometers squared of land in the 2020 year of the outbreak. And in the 2021 year, there were 9 to 15 trillion caterpillars involved. Now, just to give you a sense of how many of these caterpillars there are, we know that there have been about 350,000 kilometers squared of total defoliated land in the United States between 1970 and 2019, based on US government data in trying to track and control the spread of this animal. So if we imagine that these larvae are about 10 millimeters in size, a chain of all the US spongy moth larvae that experienced outbreak conditions would stretch to the moon and back sometime between 9 and 100 times each year, depending on exactly how dense we think each of those kilometers squared of land was going to be, probably somewhere closer to the 100 times and less close to the 9 times. So again, just a lot of animals involved in these outbreaks. There are methods of control that are applied by humans. One of those is BTK, which is a toxin that I mentioned to you earlier very briefly in the pesticide part of this. So these are biological toxins that enter the animal through consuming material that has the toxin on it. The mechanism of action is a little bit unclear from what I can tell still, but the kind of proposed mechanism is that the toxins rupture the cells lining the gut of the animal, and that causes an infection of the animal's body cavity, essentially sepsis. And so the insect is unable to eat, unable to process nutrients, and dies from either starvation or the infection within about 2 to 7 days. So BTK toxins are sometimes used to control spongy moth outbreaks, especially in areas where there might be, trees that need to be protected for economic reasons or for heritage reasons or biodiversity reasons. But actually, largely, spongy moth control is through natural issues. So the US government estimates that about 80 to 90% of the animals in a spongy moth outbreak will die by starvation, predation, and natural disease in the second and third year of the outbreak. To give you a sense of some of those natural issues, some of which are actually also being harnessed for control purposes, one is a viral pathogen called NDNPV. So this virus causes really high mortality when larvae are at high densities. It's thought that this is partially because of all the larval competition and stress, essentially increasing the likelihood that their immune function is not very good, and so they're more likely to die of this virus. The main symptom is called larval melting. That's liquefaction of the larval organs, which begins prior to death. That usually occurs within 5 days in the lab, but we don't actually know that lab conditions accurately reflect field doses at all, and so it's unclear how long this could take for larvae in the wild. We do actually sometimes spray LDNPV as a method of control, but it also exists naturally in the environment for these animals. Another pathogen that was actually introduced intentionally to the United States, again, as a way to try and control this animal. And so it is not deployed in like a case-specific basis, but rather is just naturally in the environment now, is this fungal pathogen. This fungus essentially eats away at the insides of the caterpillar and then explodes out through the exoskeleton to release the spores. This takes between a few days and a week to kill each individual. In the photo on the right-hand side of the screen that you're seeing, this is from a 2024 spongy moth outbreak that occurred in Wisconsin. So you can see how some of the caterpillars are sort of just straight lines hanging down. Those are ones that had the fungal pathogen. The ones that are in a U shape are the ones where their insides liquefied, and so they are the ones that died from the viral pathogen. So you can just see kind of the density of the effects of these pathogens in the environment. Okay, so what could we do about invasive species management and the spongy moth? Stopping spread to new areas, probably really key to reducing welfare issues by reducing the number of areas in which outbreaks can occur. And if we were better able to predict outbreaks, that would lead to better prevention strategies while the animals are still eggs. And thus, there would be a lot less potential suffering as a result. So it's my view, at least from the research that we did in that case study, that technology that reliably kills eggs, sterilizes adults in higher density populations, and predicts where outbreaks will occur so that those eggs can be targeted for eradication will do more to prevent outbreaks than trying to change outbreak management, because only about 10% of the animals are actually being affected by anthropogenic management in any case. Okay, the last case study that I'm going to talk a little bit about is actually a much smaller scale case study, but it's one where there is a lot of direct human intervention, and so there might be ways that we could again change our practices. So entomologists collect large numbers of insects. We want to understand biodiversity declines, range shifts, and more. And so we collect insects in the wild as a way to understand where they occur, which species are occurring there, what is causing them to decline, et cetera. Frequently, we use passive traps for this, like the two that I have on this slide. One is a malaise trap, which essentially intercepts all flying insects. They then crawl to the top of the trap and fall into a little container that has a bunch of liquid in it in which they drown. Another example is a beeble, like you can see the blue, yellow, and white beebles on the screen. Again, this is something where the animals are flying over it, think that it is a flower. It is full of a liquid that they then drown in. These traps are used all over the globe to understand insect biodiversity. They result in billions. We actually don't have any estimates, but it should be in the billions of dead insects each year. And that includes both target insects, which are the ones we're actually trying to study, like pollinators, but also bycatch insects, which are ones that are not of the target group, but are caught because it's a passive trap and thus aren't actually studied at all and contribute very little to science, but are dying anyway. When we looked at kind of reporting on insect welfare and ethics and reduction practices and things like that in entomology over a 20-year period, we found that essentially 0% of papers had ethics statements in most of the entomology journals we looked at, other than animal behavior, represented by AB on this graph. But to give you a sense, two of the journals that are most commonly going to be publishing on people collecting wild insects would be Insect Conservation and Diversity, 0% over the last 20 years in that publication, and in ecological entomology, only 1% of studies. So what this suggests is just that our discipline because I'm an entomologist, right? We may not be thoughtful a priori about the possible welfare harms of our research practices when collecting insects in the wild as a part of our effort to understand biodiversity. It is true that this is a topic that our discipline is beginning to talk about more, especially in the pollinator community, I think. Because pollinators are cute and fuzzy, they've been perhaps something that we've started to think a little bit more critically and carefully about public perception, and that has led to people thinking a little bit more also about welfare harms. So there are some papers just over there on the right-hand side of the screen about asking people to be a little bit more cautious in how we monitor for insects and when we monitor for insects. It may be true that lethal collection is necessary for certain research aims. I think you could have a whole discussion about, when it's necessary, whether those research aims are as valuable as they're intended to be, and more. But I also think that even without arguing about that particular point, we could still advocate for more thoughtful approaches to collection. For example, we could ask people about whether they could replace collecting new specimens with using old specimens that are already in existence at museums, if they could replace using live samples with things like photos or catch and release for identification of animals that you do actually need to handle and manipulate to understand their species. And certainly we could ask people to consider reduction more. So could we reduce the number of animals we're actually capturing by using active methods to reduce bycatch, or reducing our site coverage when we're doing studies? So maybe we don't need to be assessing as many sites or using as many traps at each site. I think in general, there's been a conversation in entomology moving towards the use of fewer lethal methods and, towards more non-destructive methods. These are just two examples. One of these is a YouTube video from the Insect Welfare Research Society's seminar series from two entomologists who have been talking about the needs and means of using non-lethal methods in entomological research. And they actually have also published a paper of the same title in the Annual Reviews of Entomology last year. There was a recent webinar hosted by the Insects Journal, also on the use of non-destructive methods, that you can find a recording of if you're interested in any of the specific methods that people are actually using to try and reduce lethal methodology. Also, this website is an incredible resource if you're a wild animal researcher working on any species. It is not just on insects, but it's about essentially methods for, again, replacement, reduction, and refinement when working on wild animals. And it does have a whole section dedicated to invertebrates. We also do have now the guidelines for protecting and promoting insect welfare in research produced by the Insect Welfare Research Society. Those include information on refinement, less on replacement and reduction. So we talk about things like considerations for sampling and collection, using active methods over passive methods where you can, husbandry, transport, invasive methodologies, anesthesia, analgesia, euthanasia, and release. All of those are covered in our guidelines. Okay, so those are the three case studies, and I want to end with a very brief note on some of the arthropods that we didn't talk about today, but that are actually even more numerous in the wild, the mites and the springtails, which are, I've put a lot of pictures of them up here for you. This is, again, very diverse groups of animals. Springtails are essentially ancestral hexapods, so they're in the group with insects. Mites are in the chelicerates, so they're in the group with like spiders and horseshoe crabs. Often, again, very small animals, although often like, people think they're all small, and just like with insects, they don't all have to be super small. But also they're even more abundant than insects are. So this graph over here on the left is showing you my estimate of insects on cropland using the 1,000 to 3,000 meters squared estimate. So that blue bar is all the insects on cropland that are being affected by pesticide sprays. And I'm comparing it to the number of fish we catch in the wild, the number of crickets we farm, and the number of chickens we farm, just to give you a sense of the scale of the individuals measured in billions in each of those contexts. This right here is a graph showing you how many mites and springtails are on cropland, if we estimate by the numbers that, again, are used by meters squared for each of those two species. Again, rough estimates, but what this is meant to show you is just that there are a tremendous number of mites and springtails per meter squared of soil. And so we should expect lots of these animals, if they are sentience candidates, to potentially also be harmed by some of the practices that I've discussed today. Okay, so if you've fallen asleep and you're waking up right now at the end of my talk, a couple of major conclusions for you. There are a lot of wild arthropods. There are a lot of insects. There are even more mites and springtails. We didn't even talk about any of the non-terrestrial arthropods. So if we did, we have even more wild arthropods to be considering. At least some of these animals are plausibly sentient. So being cautious can prevent potential moral harm while we're still uncertain. And, suffering, it may be a result of natural processes or a result of direct human impacts. There may be tensions sometimes between wild animal welfare and other human aims, but I also see areas of alignment. And it's very clear that we need a lot more research to mitigate harms to these very many animals that are out there. And with that, I'll just thank all the folks who contributed intellectually and scholastically to the work that I shared today, as well as to Rethink Priorities, who funded me to work on that spongy moth case study. And I would take any questions.
Okay, thank you so much, Megan. That was a fantastic talk, extremely clear, extremely informative, and really, really, really interesting. So thank you. And we already have some questions and comments coming in on the Q&A tab. As a reminder to everybody, please do feel free to ask a question, make a comment. You can do that on the Q&A tab. You can also upvote, interact with other questions, and then Megan and I can have a conversation based on your contributions. So with that in mind, I will start with the most upvoted question so far, and this is from Mal Graham, who asks, how much do we know about the lives of insects who don't reach adulthood? Curious whether it might still be enjoyable to be an insect if you don't reach reproductive maturity.
It's a great question. Juvenile insects are more poorly studied than, in many cases, are more poorly studied than adult insects. I think part of the reason for that is just because since they go through so many instars, which are essentially the developmental stages of an insect, it's really hard to track those instars sometimes, and so it can make it difficult to study them, and that makes people want to study adults that are fixed at a developmental stage. So in some senses, we know very little. I guess I'll give you some, again, wildly speculative thoughts on, what I know about juvenile insects and how they might apply in the wild. So one thing would be that we know that some juvenile insects have preferences. There was a really interesting study that came out recently on yellow mealworm larvae and kind of goal-directed behavior, where it was very easy to train them to move towards rewards and away from non-rewards, where the reward was an apple slice. And so you might imagine, at least I think very potentially naively about my own preference for, say, like, I don't know, chocolate. I have a preference for chocolate. That brings me some amount of joy. It seems plausible to me that insects may be able to get some positive affect out of some of the things that they find in their environment, especially nutritionally. Because they're juvenile insects, they don't, like you mentioned, they're not reproductively mature, so they're not going to get pleasure out of things like sexual activity or something like that. But we could potentially say that there are environmental conditions and nutrition that could still, allow for behaviors to be expressed that would be positively effective for them. Of course, we might also expect that there are many things that might bring about negative affect for them. So, for example, predation and parasitism, those are both pretty common in juvenile insects. There are wasps that specialize on juvenile insects, so they lay their eggs inside the body cavity of the juvenile insect. Those eggs hatch and eat them from the inside out. Seems plausibly negative in affect to me as an experience. So I think we don't have good data on a lot of insects of how many actually reach reproductive maturity, how many are affected by starvation, parasitism, predation at the juvenile life stage. And it's going to vary significantly, I would say, by species or by group. Where, for example, praying mantises, there are some species of praying mantises where very few of them are going to reach reproductive maturity. They cannibalize each other. It's hard for them to catch enough food. And so the expectation is that a lot of them are going to starve to death before they reach adulthood. On the other hand, there are some species of flies where they, have an average of 3 offspring over the course of their life. And so for those species of flies, you would expect a large number of them to reach reproductive maturity. So again, hard to say things about insects as a group just because there's so much variation. I guess the last note that I'll say about immature insects is that we do have two and then a third that I won't talk about. We have two major categories of developmental strategy. We have hemimetabolists and holometabolists. So the holometabolists are the ones I think people tend to think of when they think of immature insects. These are ones that start out like a little larva grub type form, like a caterpillar. They go through a complete metamorphosis pupation period where they're in the chrysalis, and then they emerge as an adult that is radically different in form and morphology and function like a butterfly. So that's holometabolists. Hemimetabolists is incomplete metamorphosis. If you think about crickets, they start out like a little tiny cricket and they become a bigger and bigger and bigger and bigger cricket until they're an adult cricket. We might think that there are very different juvenile experiences between those two different developmental strategies. The brains of hemimetabolists, insects like crickets, are much more similar to the adult brains in the juveniles than we see in the holometabolists insects. And so we might expect that the kinds of experiences or the kinds of affective states and the intensity of the affective states could be different between insects that have these two different developmental strategies.
Excellent. Thank you very, very much. I am now going to ask a question from Devin Kalish and combine it with a follow-up question based on your answer just now. So Devin asked, do you think in general hive insects are more likely to be moral subjects or sentient, et cetera, than non-hive subjects? And then the follow-up question that I want to combine that with is, so Mal asked about whether it might be enjoyable to be a juvenile insect, but of course we can also ask whether the evidence for sentience in the first place is different for juvenile insects. And I know that this is relevant on the farmed insect side too, because for example, many farmed insects are black soldier fly larvae or other juvenile insects. So both with the hive non-hive distinction and with the adult juvenile distinction, can you say anything about whether the evidence of sentience in the first place looks different?
Yeah, so on evidence for sentience, if we're using the Birch framework, so I'll start with that. We actually have less evidence in the hive insects than we have in the non-hive insects. I don't think that's because hive insects are less likely to be sentient. I think it's because we've just studied fruit flies and cockroaches in particular for pain research purposes. And so we just have a lot more evidence for sentience qua pain in those two groups of animals than we do in the hive animals that are in the Hymenoptera. So it's interesting because the hive insects, the hymenopterans, like the ants, the bees, the wasps, I assume is what we're talking about here. Those are groups that people tend to study for their cognitive sophistication. And so we know they have all these really cool, sophisticated cognitive abilities, but cognition doesn't necessarily equal sentience, right? Things can be cognitively sophisticated and plausibly non-sentient. So it may be that that evidence for cognition is more compelling to people in getting them to empathize or care, but isn't itself evidence for sentience. On the evidence for sentience we have better, stronger evidence in fruit flies and American cockroaches than we currently have in bees, although the bees are very close behind. Um, that said, once you think that something is a sentience candidate, you might then re-bring cognition into the conversation because it can bear on the welfare experiences an animal might have. So we might think that there's things about being a social animal versus being a not social animal that bear on the kinds of welfare experiences you could plausibly have. Social isolation, for example, might be unbearable for social animals and not even really particularly noticeable for the solitary, quote unquote, solitary insects. So I think the cognition is still relevant. It is not itself better evidence. I don't think there's any reason to suspect that the eusocial insects are more likely to be moral subjects than the non-eusocial insects at this time. the evidence for juveniles and adults, you're right, there's very different evidence between those two different groups. And actually, I didn't put it in my slideshow presentation for time, but we did split the Gibbons review out by adults and juveniles, and actually even split the juveniles where we could by early and late instars. Um, so that was our attempt to get at this question because, especially again in the holometabolous insects, their neurobiology is radically different between the juvenile and the adult life stage. So essentially what we found there is that, for Drosophila melanogaster, which is really the only juvenile insect that anybody has studied at all for any of this stuff, because again, people just don't study juvenile insects. The main takeaway of the juvenile insects table is nobody has looked for any of this in the vast majority of juvenile insects. The one juvenile insect that again gets used for pain research is Drosophila melanogaster fruit fly larvae. If we look in that group, they met 4 of 8 criteria to a high or very high degree of confidence, with the other 4 showing up as very low, no research found. So it's enough to be nervous, I guess is what I would say, right? Not as many as the decapods, but also I think trending in a direction where, given that 4 of the criteria are showing up as unstudied, we might want to be careful.
Excellent. Thank you so much. And we now have a question from Brandon Keem, which is in some ways a follow-up on or a request for expansion, elaboration on your answer to Mal's question as well, which is might already have discussed, but a question that dovetails with Mal's. Can you talk about the sources of positive experience in insect life? So broadly speaking, where can insects find positive welfare in the wild?
Yeah, again, great question about which there is essentially no research. So I'm going to speculate wildly for you. Um, if I were looking to research positive experiences in wild insects, I would look at nutrition. So are there nutrient sources that are, dense, that are preferred, that they enjoy? Especially by looking at what they choose to eat when they have choice, when they self-select their nutrients. I would look at environmental conditions. So insects are ectotherms. And so essentially that means that their body temperature is very closely related to the temperature of their environment. They're cold-blooded, if you'll let me use that term. I don't really like it, but that's the colloquial. And so you might imagine that there are certain like temperature and humidity conditions that are more comfortable for them or more optimal for their physiological processes that they thus might find, again, to be preferred. So perhaps there are some environmental conditions that make them experience positive states. There's some work, this is in laboratory animals, laboratory kept animals, but from Lars Chitka's lab, there's some work on play and bumblebees, and there was recently some work also on play and fruit flies. Um, and anecdotally, a report from a friend of mine who keeps cockroaches as pets, she has reported play in her population of cockroaches where they actually build them a swing and they will get on the swing and swing. So motion-based play seems to be something in flies and cockroaches if we trust her anecdotal report. And then object manipulation play in the bumblebees. So you might imagine, again, it's balls when they're in a lab, but you might imagine that for a young bumblebee, it's manipulating a flower in the field. That might be the object play example in the wild. Nobody's researched this at all as far as I can tell, but like sexual experiences could potentially be positive for insects in the wild. There are some insects that mate very rapidly, and there are some insects that actually mate for prolonged periods of time. The bee species that I studied in the desert, they actually have like a 6-minute long mating sequence that they do. And as part of that mating sequence, so the actual copulation is very short. And then there's like 5 minutes after it where the male like strokes the antenna of the females with his forelegs and stridulates. And the hypothesis for why this occurs is that it lets the female know she's been mated with so she doesn't mate with any other males, right? Like it's a mate guarding tactic. But of course, I don't know, like how does it feel for a bee to get their antenna stroked by a partner bee? Um, again, speculating wildly here, but I think it's an interesting question whether any of those kind of post-copulatory experiences could have some aspect of positive affect to them.
Great, thank you. We have a related question as well from Mark Herman, who asks you to weigh in on an issue very commonly debated these days about whether in expectation, insect welfare in the wild is net positive or net negative. In other words, do you expect, given the evidence currently available, given what is currently known and unknown, that happiness outweighs suffering, or that suffering outweighs happiness, or neutral, or totally clueless?
Yes, I'm going to unfortunately, Mark, respectfully decline this question, only because I think that I would be speculating so wildly on that it feels impossible to give a binary choice answer with any level of credence to it whatsoever. And I always worry, there really aren't that many insect welfare scientists out there. I can literally count on one hand, like the number of people that are empirically working on insect welfare right now. And I always worry that if I say something really definitive, people are going, that sounds definitive. Even if I try to qualify it with 500 qualifiers, people are like, oh, she said they've got net negative lives, or she said they have net positive lives. And they're going to take it, run with it, and it's going to inform their entire framework of how they interact with these animals. And so I think for questions that important, where the evidence is as thin as it is, I have to respectfully decline.
Yeah, I think that makes a lot of sense. And we have thought similarly about how to answer those sorts of questions or what positions to presume about those sorts of questions at the Wild Animal Welfare Program here for similar reasons. Now just a follow-up question then. Obviously, when we consider the effects of human activity on wild insect populations, a lot of that comes through direct interaction with individual insects, like do we kill them with pesticides? If so, which ones? But a lot of it comes through indirect interactions, including the effects of human activity on like insect habitat and population abundance. And so, do you kind of adopt a neutral stance right now about whether more habitat or less habitat, more abundance or less abundance is good from an insect welfare perspective, owing to your current state of uncertainty or neutrality about whether welfare is net positive or net negative in wild insect populations?
It's a good question. I think, what my answer should be is the exact same answer I just gave you the first time, which is I'm still so uncertain I can't, weigh in on the consequences of knowing whether their lives are net positive or net negative. I think because I am an ecologist by nature, by training, by disciplinary background, what I tend to think, my, bias would be towards thinking that improving, resource availability, improving habitat, would lead to positive welfare benefits for these animals in the wild. Um, that would be my line of thinking. I think you could question that line of thinking though. For
Hmm.
me, I tend to think that although, with increased biodiversity, you may also be increasing um, well, actually, I think part of what I'm going to say here is that there are ways that you could increase the biodiversity of particular categories of insects. If you yourself were somebody who was worried, like, if I increase habitat availability, is that going to, increase harm by increasing the number of parasitoids and predators at such a rate that it leads to more suffering for animals in the wild on net? I might say there are management practices where you can try to improve pollinator abundance without improving parasite abundance of those pollinators, for example. And if you took that management plan, you would be able to increase the number of pollinators, the habitat available for them, the resources available for them, without actually having the negative consequence of more parasitoids for those pollinators. So an example of this is that a lot of people have started putting up bee hotels in their yards, people who care about biodiversity. They put up these little bee hotels that have like open facing twigs in them, and solitary bees can come and nest in those bee hotels. Great for them to have that resource. Can definitely be good for, wild bee populations to have more of those resources. The problem is that a lot of people don't realize they actually have to manage those bee hotels, because if they don't, they will get totally overrun with parasites. And so then those bees are getting high parasite loads, spreading those parasites to wild bees, and that might actually on net be worse. So there are things you can do to prevent parasite abundance in your bee hotels through regular cleaning, especially during the wintertime when everyone is quiescent or still an egg. There are things you can do to manage that kind of situation so you're attracting the insects that you don't think are doing harm and avoiding growing populations of potentially harm-inducing animals.
Fascinating. Thank you. We have a question now from Miranda Zhang, who asks, if you could advocate for one specific research project to advance the state of insect welfare science, what would it be? And obviously we need a
Only one?
million. Yeah, right. You could give a top 20 if you want, but what area do you think is particularly important, particularly neglected, possibly tractable? What would be a good area for exploration for people interested in high impact insect welfare science?
I think it is going to be focused for me on pesticides. And I think you could
Hmm.
either take that on the residential pesticide front, because I think one of the most common questions I get from people who want to improve insect welfare on a personal, direct level is like, what do I do when there are pests in my house? And so I think there's actually a lot of room there, although it will affect fewer animals, a lot of room there because of consumer choice to improve how people interact with the animals in their houses and in their barns and in their yards. And so I really think that a research project focused on some of those groups of animals and the pesticides that we tend to use could make better residential pest management. Um, on the large scale, I would then just say also pesticide research, I think, is going to be absolutely essential. You know, humans need to eat things. We need to grow plants to be able to eat those plants. Um, so pesticides or some way of managing pests are going to be applied to cropland. And I think the faster we could work on making that process more humane, the bigger dent we will make in one of the largest and potentially mitigatable harms we currently do to insects in the wild, as well as, to many, many other animals through runoff and sublethal effects.
Interesting. Just a quick follow-up on that, and then we have a related question in the chat. When it comes to conducting research on, for example, more humane pesticides, how would you go about mitigating harm to research subjects? That seems like a research area where a certain amount of harm to research subjects might be unavoidable, and it would be justified as a matter of harming the many to benefit the very, 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 very many through the selection of more humane pesticides. So is that how you think about ethical justification for that kind of research, or is there a more humane way to understand which types of pesticides would be more humane?
Yeah, so I think there's two ways. So it's going to depend on your research project. Very intimately, it's going to depend on the specific research project you're trying to do. On the one hand, for things that you're trying to change that I was mentioning, like dosing or timing of application, some of those things could potentially be simulated or modeled, right? Like if we
Hmm.
have good background data, it's possible we could simulate or try to model, 60% probability of success or whatever, right? Like if we do this, 90% probability of success if we do this, hit this many target insects, this many non-target insects if we do this. So there may be some room to not use live animals when you're just talking about using the pesticide tools we already have and changing
Hmm.
how we're thinking about applying them. When it comes to developing new pesticide technologies or testing the existing pesticides to see how damaging they actually are, then I think you are probably going to run into a case where you do actually have to just harm some research subjects as a result of collecting the data. And so there you're just trying to reduce the number of total animals that you're using and refine your research practices as best you can. I think a similar thing, I do a lot of slaughter method research right now, slaughter and stunning technology for farmed insects. And it's a similar question where we can, we use methods of where we simulate, like thermal processing, for example, can be simulated if you know enough about the characteristics of your animal. Instead of having to boil 100,000 of them alive, you simulate what it would be like for each of them if you boiled them alive. Um, Other methods are not simulatable, like grinding and shredding, you cannot simulate. And so for those methods, instead, we actually are using animals and putting them, anesthetizing them before putting them through the process. And I tend to justify it, at least for myself, in a similar sense where it's, these thousands of insects in my lab are, going to be slaughtered in this way, but I'm going to produce an SOP that, if adopted by the industry, could potentially benefit trillions of animals each year.
Right, understood. Great, thank you. So the related question from Katrina Lowy is, I know that Rethink Priorities and Wild Animal Initiative are working on pesticides and welfare. Who else is researching this topic, and how can we learn more?
That's a great question. So I think there's more people out there than people realize, including me, because that's what I found when I started the Insect Welfare Research Society was that there were more like-minded individuals that we just weren't connected with each other. And so I think one thing would be just like the membership database of the Insect Welfare Research Society likely contains some people who are thinking about humane pesticide questions. And Arthropoda Foundation, if you know any of those people, is actually seeking RFPs right now or sorry, seeking proposals or expressions of interest right now. And I think they'd be open to pitches that might be focused on things like, um, you know, more humane pesticides and stuff like that. So I do think there are researchers out there. Actually, even also in the vector control case, I attended a really interesting talk at the Royal Entomological Society's Welfare and Ethics Special Interest Group meeting in 2023 from a mosquito biologist who was talking about more humane pesticide development for mosquitoes that carry malaria in Africa. And essentially, most of what was making that more humane was reducing non-target animal effects that are very common with current pesticides that are used. So I think people are thinking about it, but I think there's not like an organization that you could point to and say, oh, that's, humane pesticides are being researched by that group. But there are researchers that are interested in this topic.
Yeah, great. I can add, by the way, that while the NYU Wild Animal Welfare Program is not currently investigating this question from a scientific perspective, we are investigating it from a policy perspective. So last year, we released a policy report about how cities and other local governments can include wild animal welfare in their infrastructure and built environment policy decision making. And this year, we are following that up with a report about how cities and other local governments can include wild animal welfare in their pest and conflict management policies. And we expect to release that report in the fall. So if folks want to be in touch with case studies or ideas, feel free to reach out about that too. I think doing this kind of work at the science policy nexus can be helpful, both because it can increase insect welfare, at least to some extent, and wild animal welfare to some extent in the short term, but then you can also monitor the effects of policy experiments and iterate and improve over time. So just flagging that policy work.
I will also say, like on your point, Jeff, about policy work, like there's, I think, is a lot of room. When I read the conservation literature talking about pesticides, there's a lot of room for us to spray less pesticides than we do. We are often we're over applying, like too many doses, too many applications each year. And so there is room already, even just using the tools that we're already using without any additional research on what to do to be more humane, to just scale back on some of what we currently are doing and what we currently have. And that may be an area where some of that policy work is especially influential to say, look, we don't have a more humane pesticide necessarily at this exact moment in time, but do we have to be applying this pesticide 20 times to this field over the course of the season? Or could we apply it 3 times, and that would
Yeah.
affect a lot fewer non-target animals. I guess the last thing I should say also is that Monarch Crops is a new company that I'm actually consulting with out in San Francisco, and they're working on developing insect repellents for spotted wing Drosophila instead of insecticides, which are currently used, and IPM strategies, which are currently used.
Great, excellent, thank you. So we have a lot more questions. We will get to as many of them as possible. We might move into a little bit more of a quasi lightning round in order to make that possible, but we still have a little bit of time. So first, just a quick follow up from Mal directly on the topic of research methods, which I think is very interesting. What is the possibility of doing observational studies or quasi experiments where you monitor outcomes on farms, fields, et cetera, that are already applying various pesticides? So animals are still dying, but they would have died anyway, separately from your study.
I think that's absolutely doable. I think the main challenge you might face with interpreting your data in those cases is just that the data is going to be super messy. So you're going to have a ton of other variables that vary across your plots, and figuring out what's due to, particular pesticides being used and what's due to, other variation in your plots can be really hard in those kinds of studies. But people do still use those kinds of studies already, for other ecological questions. So it's definitely doable. And if you were just thinking of like trying to, monitor like how long does it take for an insect that is sprayed with a particular pesticide to die from that pesticide, you could literally sit in a field behind the sprayer, and like collect insects and see how long it takes. Instead of having to spray any yourself. So I think, if you had good partnerships with your farms, there are ways you could do some of the work in the field.
Excellent, thank you. We have a question from Steen now, who says, the three wild insect welfare issues you covered seem to be highly relevant in the US context. I am assuming that is because this is the region where you have data. I expect that pesticides are a concern everywhere, but do you think globally there might be other issues that are particularly relevant and I think maybe neglected or less salient within the US?
Yeah, so all three of those issues are going to be globally relevant. Pesticides are used globally, invasive species outbreak. So I just mentioned one, like the spongy moth as an outbreaking species, but that is one of many, many, many outbreaking species. And in fact, spongy moths, which are native in Europe, do actually sometimes go through these outbreaks in Europe as well, where they're native. And so this outbreak cycle that is a problem in the US and is managed as an invasive species problem is a non-invasive species, but still welfare problem in the EU where these animals are found. And entomologists are collecting bugs literally everywhere. So we just love to collect bugs everywhere we go. So it's not just a problem in the US context for that reason. And in fact, there are a lot of calls for more monitoring of insects in the tropics because we have an especially poor understanding of biodiversity and its potential declines in the tropics. So you might imagine that even though most of the studies that have currently been done with collecting insects have happened in North America and Europe, that there may be trends towards movements to the tropics in the future for entomological collections. Um, Other issues that may be globally relevant besides those in the wild insect case. One is probably harvesting of wild insects, either for food consumption or for shellac or other products. That's not super common in the US. Most people in the US or Europe even are not super into going out into their backyard, picking some caterpillars and crickets and cooking them up for dinner. But that is much more of an issue from a welfare perspective in some of these other countries where animals are more routinely collected from the wild for food consumption or other purposes. I think, you're starting to run into the like, when is it a wild animal welfare issue and when does it become like a captive animal welfare issue at that point? But it's certainly crossing that nexus, the harvesting. Um, that's all that's coming to mind to me right now is that, all of those issues are global issues and the addition of harvesting. I'm sure there are things that I'm not thinking of.
Yeah, no, of course, but that is already really, really helpful. So thank you. We are now going to careen from the hyper global to the hyper local and ask a question from George. And I want to tack on a more general version of the question. George says, isolated nests of gypsy moth larvae periodically occur on trees on our property, which we mechanically remove. What would be the most humane or least inhumane method of killing these larvae? Now, you may or may not have a direct answer to that question. If you do, please offer it. Either way, I think a more general version of this question might be something like, what do you do in your own personal life to be mindful of the welfare of wild insects who live in your community? And what, in general, can people do who might not be researchers or who might not be policymakers? What can people do in their own individual lives to kind of shift perspectives about insects as stakeholders for our decisions and maybe on the margin improve insect welfare here and there? Any thoughts about this?
Yeah. So I think, one thing to note is that the tools available to you are going to depend a lot on the specific insect that you're dealing with. So like you can mechanically remove, a nest of spongy moth larvae. It's much harder often to remove a nest of ants that are somewhere in your house, but you don't know where, and it could be in the wall. And like just put them outside. And so the tools available to you are going to be different depending on the kind of animal you're dealing with. I think the first question is like, when I think now about having, what we would call pest, if you will, insects in my life. The first thing I try to do is just be more patient in my approach to those animals. Like, can I spend some time researching this group of animals and thinking about ways to remove them more politely from my property? Um, so, like instead of putting out a bait for ants that they're going to carry back to their nest and, spread to all of their nest mates as a way to get rid of those ants, ants are coming into my house because they need food, right? So can I put food outside of my house first, still
Hmm.
in some radius where they might be likely to find it? And then also, can I clean up the inside of my house very frequently in the area where there are ants to remove their chemical foraging trails so that they're not finding food or, not even communicating as well with each other when they do find food inside my house and instead finding the resources they need somewhere else, right? So can I encourage them to move somewhere else by giving them the things they want, just not in my house? Sometimes that's not possible. Like in the case of your spongy moths, you really do want to remove them. So there in those cases, I think I always try to look for what is the fastest way that I can, kill this particular animal if I really do need to kill them. I can't just move them. So there, Honestly, if it's a small number of animals, squishing them is the fastest way to kill them. Individually squishing them. It feels terrible, to be perfectly honest, like as a person to kill them that way. But if you can completely and totally squish them flat in one solid go, that is probably a better experience than putting them in the freezer, than, boiling them, than just leaving them to die in a place where there are no resources for them, and so they starve to death. And so I think like if that's something you have to do, that is the way I would recommend. Um, Even though it can feel quite bad. The last thing I guess I'll say is that sometimes I think people look at insects in their environment and they may think that an animal is either doing well when it's not doing well or the opposite. So people have difficulty assessing the welfare state of the insect. And so they might see a bee that is like really sick and think, oh, the bee is just like a little hungry and I'm going to give it some, food and that will like, perk it back up and make its life better. And in some cases, what you actually do is just make it easier for that animal to like spread the disease back to its colony mates by giving it enough energy to like fly back to the nest. Or, you extend its suffering in other ways. When most insects are letting you willingly interact with them, they may or may not be having the best time. These are typically prey animals. And so I guess I would just say, when you are interacting with these animals, try to pay attention to behavioral signs insofar as you can of whether their lives are going well or poorly. You can always try feeding that bee, seeing if it seems like, when it perks back up, it's also behaving normally. That's really hard for people to assess who aren't bee experts. What is normal bee behavior? But you can find a lot of videos online of healthy bees and unhealthy bees, for example. And again, sometimes one of the kindest things you can do when you're dealing with like a bee that has had a sublethal pesticide exposure and it is literally trembling right in your hands, like you can leave it there to slowly die from that pesticide, or you can kill that insect and potentially make its life a little bit, the end of its life, a little bit better. Um, this is not to say you should go around squishing every bug that you find, though, just to be clear. I'm just saying, like every time you see a bee and it crawls on your hand, that is not immediately a sign to be like, oh, this is so wonderful that this bee loves crawling around on my hand. It may be a sign to look closely and see if there's an opportunity for mercy.
Yeah, interesting, interesting. Very tough. I really like the sort of general framing and foundation of the answer, which is, of course, violence should be thought of as a last resort, not as a first resort.
Yes.
You can explore these other opportunities to either relocate or to incentivize self-relocation. And you can also put up with a certain amount of nuisance or inconvenience. And
Yeah.
then when violence is truly necessary for some kind of vital self-defense, then the least harmful means, which could be squishing, even if it feels more uncomfortable. I think that is really helpful. And then, of course, getting into the weeds of the particular cases is a whole other conversation. But yeah, no, thank
And
you for that
again,
type of answer.
when insects are in your house, they want something, right? They want a
Hmm.
temperature or they want food or they want water. And if you can figure out what it is that they want, you can provide that for them elsewhere. And so that's
Right.
often, if you're willing to be patient and you can, finding
Hmm.
a way to provide that resource elsewhere can sometimes be possible.
Excellent. Thank you very much, Megan. Now we are over time, so we are going to close it here. Apologies to those of you we did not get to, but I am glad we got to as many questions as we did, and we can share the remainder of questions with Megan just so she can see what was asked and take that into account in her future work. So please trust that she will see your questions no matter what. And let me just now say, Megan, thanks so much for taking the time to talk with us. That was an incredibly interesting, informative talk, and your answers to the questions were also really helpful, wide-ranging. And thanks to everybody who attended. We had a great conversation. Really appreciate your questions and comments. I am going to give a quick shout out to Audrey Becker, who organized this call and hosted it. Thank you so much, Audrey. And to the rest of the team at the NYU Wild Animal Welfare Program. Again, please do sign up for the email list at the NYU Wild Animal Welfare Program and sign up soon because we have an exciting announcement about our program coming up over the next month or so. So you can stay tuned to the email list to hear what I am teasing right now. So thanks again to everybody for joining us, and I hope you have a great rest of the day and see you all soon.